I'm Harry Warner, and it's my pleasure to tell you about our recent short report in the Journal of Cell Science. Our study focused on how dendritic cells rewire their trafficking machinery to facilitate IL-6 secretion following pathogenic LPS stimulation. We found using turf microscopy that dendritic cells increase IL-6 secretion following short-term LPS stimulation on a time frame of 1 to 4 hours. We previously observed that the snare protein, VAMP3, is rapidly phosphorylated following LPS stimulation in dendritic cells. This was of interest because VAMP3 is essential to IL-6 secretion. We therefore hypothesized that a phosphomimetic VAMP3 mutant would better complex with its binding partner syntaxin 4 in order to release IL-6 outside the cell. We measured complexing of VAMP3 with syntaxin 4 using film microscopy, and we found that the phosphomimetic variant of VAMP3 could better complex, but much to our surprise, so could the phospho-dead mutant. We therefore hypothesized that both mutants actually disrupt an interaction between VAMP3 and its inhibitory chaperone WDFY2. Indeed, both mutants were less able to co-localize with WDFY2 relative to the wild-type variant. Furthermore, the wild-type variant of VAMP3 showed reduced co-localization with WDFY2 following LPS stimulation. In addition to this, we found that the phosphomimetic variant of VAMP3 was able to promote IL-6 secretion relative to wild-type. All these data points put together suggested that VAMP3 positive vesicles should be more polarized following LPS stimulation. Indeed, by segmenting dendritic cells using the actin cytoskeleton, we were able to measure VAMP3 vesicle polarization and saw that it increased following LPS stimulation. Furthermore, by reanalyzing our turf movies, we were able to see that IL-6 secretion is highly asymmetric and therefore polarized. Therefore, our model is that in the absence of LPS, WDFY2 holds VAMP3 in place. LPS stimulation disrupts this interaction and allows VAMP3 positive vesicles to reach the plasma membrane, and this primes dendritic cells for polarized IL-6 secretion once IL-6 has been synthesized.